A-Level Biology 9700, Paper 4, Variant 2, May-June 2025. And in this video, we will discuss the question number 8. And question number 8 is, a lichen describes a mutually beneficial association of a fungus with an organism termed as photobiont. An example of photobiont is the green alga, Treboxia species, which is a photosynthetic protoctist. Shows lichen attached to a tree. You can see the figure 8.1. Now the question regarding this picture is, outline the characteristic features of the kingdom fungi. So we have to write down the few of the characteristics of this kingdom fungi. So here there are, you can say, some of the structural features of this kingdom. So first one which I would like to write is its cellular structure. That is, it is eukaryotic. Fungi has complex cells with a defined nucleus and membrane-bound organelles, and its cell wall is made up of cellulose or bacteria, the peptidoglycan. Fungal cell walls are made up of tough polysaccharide called chitin. Because the cell walls of the plants are made up of cellulose and bacteria peptidoglycan, so they are differently made up of another type of polysaccharide, which is chitin. And most of the multicellular and consist of the network of thread-like filaments called hyphae. So mass of these hyphae is known as mycelium. So they are heterotrophic in nature, that is, they are unable to manufacture their food by the process of photosynthesis. So we can say that they have saprophytic way of nutrition. They are typically feed by extracellular digestion. They secrete enzymes onto organic matter to break it down into simple molecules, which are then absorbed across the cell membrane. So I think these points are enough, which we can write as the answer of these. So I will start writing. So the first point which I am going to write, that is the fungi. They have complex cells with a defined nucleus. Complex cells with a defined nucleus. And membrane bound organelles. Their cell wall is made up of a polysaccharide chitin. And the plants, the cellulose, and in bacteria, peptidoglycan. These are the two components of the cell wall of those organisms. But here in this case, this is the cell wall is made up of chitin. Multicellular and consists of network of thread like filaments called hyphae. And group of hyphae is called as mycelium. As mycelium heterotrophic in nature cannot able cannot manufacture the food so live a life of saprophytic organism. Saprophytic organism and secretes enzymes onto organic matter to break it down to absorb the nutrients. So it was all about the part one of this question. So now the part B. So part B suggests how a fungus and a green alga benefit from the relationship shown by the lichen. So you can say that the lichen and the green alga exist in a mutualistic relationship where both organisms provide essential resources to one another that they could not easily obtain alone. So the benefits are typically divided. Many benefits are there. So one of them I can show you that this is a mutual relationship or symbiotic type of relationship in which both of they give benefits to each other. So one example which we can say that uh, you know the fungi are heterotrophic in nature, so they cannot produce their own food. So as a result, the green algae performs photosynthesis to produce organic uh, carbon compounds to which they and then transfer to the fungus as a primary energy source. On the other hand, the fungal hyphae physically surrounds and encase the algal cells, protecting them from her harsh environment. So this can be the example that how they benefit to one another. Number one, symbiotic relationship is between them as fungus is heterotrophic so get food from green alga and as a result provide protection to green alga. And also provide moisture moisture and minerals. So in this way, they benefit each other. Next question, part C is there. A suspension of triboxia in water was used to investigate the effect of the intensity of light on the rate of photosynthesis. The volume of oxygen released over a set period of time was used as a measure of the rate of photosynthesis at each different light intensity. All other conditions were kept constant. So shows how light intensity affect the volume of oxygen released by triboxia. So here you can see figure 8.2 shows how light intensity the volume affected the volume of oxygen released by triboxia. And here you can see at 0, less A is less than 0. So it means there is no photosynthesis at that time because light intensity is 0. And it means the rate of respiration is high over there. Only the respiration is taking place at that time. So when light intensity after this at point B, both of them becomes equal to each other. And then as the movement is from B towards the C, you can see at C, the rate of photosynthesis is reached to its maximum. And then it becomes levels off. It means uh, at that time, now more oxygen is volume is produced is same. So it is level offs. So now one by one, I would like to describe the answers right over there. State which photo system is involved in the release of oxygen. So in the release of oxygen, in photosystem 2 is responsible for the release of oxygen. It contains a specialized structure called the oxygen evolving center where photolysis occur. So in this process, light energy is used to split water molecules into hydrogen ions, electrons, and oxygen gas, which is then released into the atmosphere. So photosystem 2. So photosystem 2 or PS2 is responsible to release oxygen because photolysis takes place when photosystem 2 absorbs the light. Now the next question is with reference to figure 8.2 already explained the answer. Explain the curve between A and B and explain why the curve levels off after C. First of all, I would like to write at point A, there is zero light intensity. At point A, light light intensity is zero. So only respiration is taking place. No photosynthesis. And as we move from A to B, the light intensity increases, causing the photosynthesis to occur. In this region, light intensity is the limiting factor. At point B, the rate of photosynthesis matches the rate of respiration. And oxygen release is zero. And the second part of this question is after point C, the rate of photosynthesis reaches a plateau. That is no further increase in the rate of oxygen release. As light increases. Now light will not be a limiting factor at this point. And enzymes are working at at their maximum possible rate. Wait, no. We can say that this is the saturation point. So it was all about the question number 8. Please like, share, and subscribe my channel to watch more videos. Thanks for watching.